Mitchell, and I am Watershed Education Manager for Anacostia Riverkeeper. And one of my wonderful programs I run is our Community Science Project, where we get to work with you guys, amazing and interested community members, to monitor the water quality to share with your neighbors. And so today's training is going to walk you through our programming and get you ready to be a volunteer this summer. Give me 1 second to share the screen. We'll get going through these presentations. All right, so volunteer water quality monitoring. We want to start by just thinking that we have a lot of funders and partners helping to make this happen. And so we, of course, are thankful, and our Montgomery County programs are funded through CBT grant, funded by MoCo DP and the MoCo Montgomery County government. So we're really thankful for that funding to allow this work to happen. We also have a lot of amazing community members and partners that help kind of recruit volunteers and get the word of water quality out there. So Neighbors of Northwest Branch, Friends of Sligo Creek, Defensores de la Cuenca, One Montgomery Green, Joe's Movement Emporium, Friends of Lower Beaver Dam Creek, Sustainable Heightsville, and Friends of Quincy Run probably brought you guys here. They probably will also share this with you to know to take this training. So we're really thankful for their expertise and their assistance with this project. So I want us to start by thinking, why monitor urban streams? Isn't the water perfect and pristine and beautiful and clean? I'm sure a quick brainstorm, you're recognizing that there are a lot of things that might make the water not quite healthy. Road salt, we know, is one of the things that can kind of mix with the chemistry of the water. That chlorine levels get high and starts messing with the natural cycles. We know that storm debris and storm water, including leaves and extra detritus and nutrients from fertilizing in the water. We know that motor oil and pollutants that both are point and non-point type pollutions that get into the streams. We know there's issues with litter, which is not only unsightly, but can break down into microplastics that harm the watershed. And then one of the big ones we talk about in urban areas is the fecal bacteria. We know that there's all types of animals, unfortunately, in the area that poop, both natural ones, birds, geese, deer. They're supposed to be here. They do poop. So some fecal bacteria is normal, but we also know that there are lots of more dogs, sometimes irresponsible dog owners that don't pick up their own dog species, as well as improperly connected human wastewater systems that can lead to excess fecal bacteria in our watershed. And so in this project, at Anacostia Riverkeeper and with our partners, there are a lot of things we monitor, but the ones we focus on in this community science partnership that you're part of is looking at some recreational standards, really telling people if the water is safe to be in contact with. And also just saying some really broad stroke measures of stream health, letting us know where we might need to put more work into. So with recreational standards, I'm mostly, again, talking about that E. coli. That's one of those fecal bacteria that we know could make people really sick. So E. coli itself can make you quite ill if you accidentally ingest it. But it's also indicator bacteria. So studies have shown that if E. coli is high, the other stuff might be high as well. We look at turbidity. That's how much sediment's floating in the water, how dirty or cloudy that water seems. That's also going to help us with some stream health, knowing if there's excess sediment in the water, sunlight's coming through, aquatic life can't thrive. Looking for pH, making sure nothing's too acidic or basic is happening. Monitoring stream temperature as climate change and decreasing tree canopy lets more sunlight hit those streams. We're noticing it creep higher, so it's important that we record that. And then this year with the Smithsonian Ecological Research Center partnership, we're doing some really cool look into fertilizer and nutrient cycling throughout the water system. So focusing on nitrogen and phosphorus, the popular ingredients in fertilizers. We know that a lot of residents might use on their soils. It's highly regulated, but are those regulations being followed? This study will help us get a better glimpse on that. And again, especially with that fecal monitor, it is not just something that we want to avoid, because we know for a fact that it can make people, and it has gotten made people who recreate in the region's water get sick. It's a great indicator monitor to know if other of those pathogens might be around. So when it rains, all of those pathogens, I'm looking across my street at a huge parking lot full of geese. Obviously, geese are natural, but probably that many geese in one place, there definitely is some human influence there. And as stormwater washes away, if it's supposed to rain heavily tonight, it's going to take all that poop the goose put on that parking lot, and it's going to put it in the stormwater drain. And from that stormwater drain, a lot of people think that that water is going somewhere to be treated, but that is not true. Those are going to go to outfalls, which are just big culverts that dump that water directly into your local stream or brook or creek or river. And so knowing that that's where that water goes and knowing all those things that might be on the parking lots and the lawns in the neighborhoods, we want to make sure we're monitoring to make sure the water is safe. So in our Upper Anacostia monitoring project, it's a bi-weekly project, meaning that we do it every 2 weeks for 10 weeks from about mid-May to mid-September. And we have a total, I've almost lost track, I think it's 14 sites. And so a lot of them are based in Montgomery County, including 4 along Sligo Creek and 2 along Long Branch, which feeds into Sligo Creek. We do have 4 across Northwest Branch in both counties, 2 in Montgomery, 2 in Prince George's County. We have a site up in Paint Branch, really close, borderline on the University of Maryland campus. Another one on Northeast Branch, just below campus, to kind of see how that area of College Park might be affecting water quality. One on Quincy Run, one on Lower Beaver Dam Creek, and then one in Bladensburg Waterfront Park. That's on the river itself. So now that you've learned a little bit about why we monitor and where, let's start talking about what it actually means to be a monitor. So this is a bi-weekly thing. So every 2 weeks, starting May 15th and going in through, I think it's about September 18th, have to look at a calendar. This process will repeat itself, right? So early in the week, you'll be signed up to a spot. We'll reach out to you, say, hey, yes, you're confirmed. You're monitoring this week at this location. I'll show you how to sign up in a second, but you'll pick a site to monitor. Maybe you'll monitor that site every single week for the entire summer. Maybe you'll monitor every week this summer, but try a different place each time. And maybe you're pretty busy. You can only monitor once or twice, and you'll just sign up for those weeks you know you're available on those Wednesday mornings. So Monday, Tuesday, you'll receive a confirmation from us saying, yes, you are done. You are signed up. You're good to go. Here's your information. And then you'll need to pick up your cooler with supplies from us. Once you get that cooler with supplies from us, and I'll talk about where to get those, you're good to go on a Wednesday morning. So sampling occurs between 7 and 11 a.m. on Wednesday mornings. We do have that as a coordinated effort because the bacteria bottles are actually quite time sensitive and finicky. So we want them all collected at once. Which allows us to have a really great grasp of what's going on across the stream at the same time, across the watershed at the same time, as well as we do have to process those samples. So having them all come in the same time allows us to process them more efficiently. So you'll monitor in that morning, dropping them off at a drop-off location, either our lab in DC, or we have a couple up in Montgomery and Prince George's County closer to you. We might postpone until Thursday with extreme weather conditions, but that has not happened the past 3 years. So just have that in the back of your head that that's a possibility, but a very faint one. And then by Thursday afternoon or Friday, we'll have our results published. Once we get the bacteria bottle, it takes me 24 hours to process it. And then a couple extra hours to put a nice visual together to share it on social media for you to see your results. And then share those results with your own community to make sure everyone has access to data to make safe decisions. So first up, I said you sign up. As soon as you completed this certification that you're working on right now, I will send you your sign up sheet. We'll have all 14 sites. Only 3 are on my little clip right now in the screenshot, but you'll have all 14. And you'll notice that 2 people can sign up for each day at each site. We do recommend monitoring in pairs. A lot of the monitoring, just as nice to have one person recording data while one person measures data. It allows you to be more efficient. Some of the work can be a little tiring. Also, sometimes stream banks are slippery. So we recommend, highly recommend monitoring in pairs, but monitoring on your own is acceptable if you are comfortable with that. So when you're ready to monitor, you'll sign up at the site. So if I'm Maureen Mitchell, I look like I am going to next Monday at Long Branch, LB1, that's near the Rolling Terrace Elementary School. So I'll put in my name, my email, my phone number. Those aren't my real emails and phone numbers, so don't try to reach out to me there. I'll give you my real stuff in a little bit. I will make sure I put that information down so real Maureen, the monitoring Maureen, the ARK employee Maureen, can give me a call or email if anything comes up that we have to do a weather date, if something's wrong with your sample and I just need you to help remember what site you're at or something. I'll have your information there. I'm going to indicate where I want to pick up my supplies. Again, you could pick them up in our DC office, but we have some satellite pickup locations up in Maryland closer to you. You're going to indicate what approximate time you're going to sample at. That will just help us with logistics if we're trying to have someone meet you, if we might need to be delivering you samples or something. Nice to know when you're doing it, where you're going to drop off. Again, you can drop it off in DC, but we have some satellite locations. And then if you're available on Thursday, again, we have not had to cancel the Thursday for couple of years, but it is a possibility. So if you're not, that's perfectly fine. That just means that I might need to be more ready to go out to those sites. Once you're signed up, notice that I put some X's in that second row after my name. That's because I'm not feeling friendly today. I want to monitor by myself. If I wasn't feeling friendly, I would just leave those X's out and someone else could fill in their name. I would have their contact information to be in touch and say, hey, we both signed up to monitor Long Branch. Want to meet at the site at 9 a.m.? That's all we have to do. It looks like Quinn and Petra above me both signed up together. Let's assume they're friends. They're both certified. As long as one person is certified, you can bring a friend along that's uncertified as long as you're the one taking the samples. But again, we really want two people to go out. I'm telling you, I've done it a lot. It's a lot easier in pairs, but you can do it by yourself if that's something you feel most comfortable with. Once you're signed up, you'll have to pick up your supplies, right? Our coolers are located in our DC office. I didn't even put that on that slide because that is far away for most of you. So most likely you're going to be choosing to come to my house. I live at 2005 Dennis Ave. It's closest to the northern or upstream-most Sligo point. And so I work here. I have a nice place in my driveway. I can leave coolers for you to pick up at your convenience. We also have a partner, Joe's Movement Emporium in Mount Rainier. And their building is open during normal business hours. I can leave a cooler there to pick up. So all your supplies are provided. We will ask that you just provide some ice. I can give you an ice pack if you request it, but you'd have to make sure you put that ice pack in a cold place between when I give you your coolers and you go out and sample. And if you have signed up with a second person to monitor, only one of you has to take this step of picking up supplies. So once you've picked up supplies, you know who you're monitoring with, you picked a time to monitor together. On the side, you're going to head into the field. Again, just keep your eye on your email, especially the night before. If I'm seeing heavy weather, potential thunderstorms, tornadoes, tropical storms, I'll be in your inbox advising you on if we're pushing to Thursday or what precautions I'd like you to take. You also will have a manual in your cooler, which will have exact coordinates, exact lat lawns of where that sample is to be taken. So rather than just heading over to Rolling Branch and looking for the creek, you can plug in the coordinates into your phone or use the images and written directions provided to know that you're sampling the exact correct location. If for any reason you're out there and you're like, these coordinates are bringing me to a weird spot or my GPS isn't working, you just hop on the phone and call me. My phone number will be in the manual as well and all the emails I send to you. And I can help direct you to the site at that time. But it's really imperative that, again, you're sampling in that correct location. You're using your manual to make sure you're in that right spot. And you're keeping your eye on your email for case I need to reach out if anything is changing about monitoring last minute. So you're at your site, you have your supplies, it's time to collect your data. You'll collect them in this order. Your starting sun temperature and pH. Those are just quick ones you'll do and record on your data sheet. Then you'll be grabbing a bunch of samples, bacteria, turbidity, which are the ones that I process, a couple nutrient samples, which our partner at Smithsonian processes, and then potentially a couple of you on a couple of occasions might grab one more sample for microbial source tracking, which is processed in our University of Maryland Partners Lab. So air and water temperature thermometers look just like this. They're the thermometers you've seen a million times. They're pretty simple. They have a sheath on that just protects that metal rod, which is the actual active thermometer. You'll remove the sheath. The blue button will turn it on. It should automatically load to be reading in Celsius. If for any reason it loads into Fahrenheit, I'll notice the number looks really weird and do the conversion myself at the lab. Don't stress. I can usually tell if you accidentally did it in Fahrenheit, but we do prefer you write that data down in Celsius. That's the way of science. You're going to take your air temperature first. You're going to try to find a shady spot, avoiding direct sunlight because that will reflect on the metal and give you a weird reading. So ideally, you're finding a shade. And the thermometer will take maybe like a minute or two just to bounce around, especially if you had it in the cooler with the ice pack. It'll take a couple of seconds just to a couple of minutes even. So just turn it on, place it on the ground in the shade, and start recording some of your data for a minute or two, allowing that thermometer to warm up from the cold cooler, and then record your data. After you do your air temperature, you'll repeat the process, but this time you're just sticking the middle tip of the thermometer into the water. Don't bury it into the sediment. Try to get it nice in that nice wet part, not the bottom, not the top. We go for mid-column of the stream, the middle. Same thing, it won't have to equilibrate quite as long because it already equilibrated to not being in a cooler while you did the air temperature. But it is imperative that you do water temperature second. And that's because if you did water first and then air, there's something called evaporative cooling. So the wetness on the thermometer after you dip it will be evaporating, and that will artificially decrease your air temperature and give us some false readings. And so air first, allow it to equilibrate while you just kind of get yourself settled. And then you can do water temperature. It shouldn't take as long to equilibrate because it's already been out of the cold cooler. After you did your temperature, Resheath your thermometer. There should be a little chem wipe or some kind of cloth, paper towel, if you will, in your bag, so you can wipe off any moisture off your thermometer so it doesn't rust between now and its next use. And stash it right back away in your cooler, grabbing now your pH strips. I'm sure you've seen these before. Oh my gosh, I feel like every chem lab in high school ever, I taught school for a long time, use these a lot. So sure, you've had a chance to use them before in your life. They're really simple strips. You'll have a little tub that looks like this. We use the Hydron wide range indicators that will make everything from 0 to 14. So 0 being the aesthetic and 14 being the basic side. We're shooting for the middle. About a 5.5 to 8 is what I'm happy with. And so you're going to grab it, holding it by the white tip. Hope you can see my cursor move right now, but you don't want to touch that little peachy orange pad. That's where the actual science takes place. So try not to touch that with your hand. At this point, I recommend putting on gloves. You'll have nitrile gloves provided in the bag. That's just because it's a chance now you might be touching the water. And the whole reason we test the water is it might be polluted. So go ahead and glove up. And then holding it by the non-peach pad side of the strip, you're going to dip it in the water. Really let it sit in the water for up to a minute, really letting the little paper pad at the end of your pH strip absorb the water, the little pH chemical reactions occur. And then you actually really want to sit it. I like to put it right on top of my red cooler, maybe on top of my data sheet with the peach side paper pad up. And it will take about 2, maybe up to 3 minutes, depending on the temperature that day. It goes faster in a warmer day. For it to kind of finish its chemical reaction. And so the true pH color will show. Using the color key provided, again, you're going to color match and estimate that pH. You're allowed to estimate to half. So if you feel like it's really between a 6 and a 7, writing 6.5 is fine. If for any reason when you pull out your strip, it's not showing that peachy color, sometimes they'll have a weird reaction where they look black. Please throw that strip away and use a different one. If you have any reason to suspect your pH strip didn't work properly, you should have plenty in your kit. So go ahead and just do a third pH strip till you feel like you're getting a result you're comfortable with. Make sure that strip, once you're done recording, never ends up back in your cooler or somewhere in a trash can, not littered into the stream. And then you can move on to your next sample, which is your bacteria sample. Your bacteria sample will be in a 100 milliliter plastic bottle. It will have this little paper strip on it. You want to make sure that strip has not been broken because that is a sealed, sterile bottle. You'll also notice inside that bottle, there's like a little white powdery substance. I am not going to even try to pronounce the chemical name of what's inside there. But that chemical is basically going to fix the bacteria. It's a preservative. So once you take that sample, it will help make sure the bacteria doesn't do anything funky in the time between you taking the sample and me back in my lab processing your sample. So we want to make sure that powder stays in the bottle and anything that touches the bottle is sterile. So at this point, you're wearing your gloves for sure if you haven't put them on already. So once you have your bottle, you can go ahead. I like to just write my site name. So if I was at LB1, I'd write LB around with Sharpie right at the top just to make sure nothing gets clear. Then you can break as long as you have gloves on, break the seal and approach the water. And you're going to take a nice scoop of water, filling that to the brim. There is a 100 milliliter line. If you look right where like the T in collector is, you can kind of see that line on the image there. That's your 1 ml line. I need to have that much water to process. Overfilling is okay. I love a good overfilled bottle. I can deal with that back in the lab, but underfilled bottles will give me some false results. And so I really do need to have at least 100. So in one nice scoop, you're going to fill it, getting it to that 100 milliliter mark or higher. Then you're going to recap. Again, I have a paper towel, so go ahead and wipe the outside of that bottle down so it's clean and doesn't get you sick. You'll have some little sticker labels that will have places for you to write your name and date and stuff. You'll stick that on, putting a piece of tape on if you feel like it's not going to stick because the water was a bit wet, and then stash it in your cooler, recording all the information on your data sheet. And again, that label looks like this. You'll have your site ID. I went to Long Branch at Rolling Terrace, so I'm writing LB1. That's my site code. You'll know your site code depending on when you're at. You write your initials, the date and the month, time. We'll do it the American way, please. And then time, you write the time you took that sample. Make sure that is the time you actively took that sample. It's really imperative I have the correct date on that, exactly what time you took that sample, because that will help me prioritize how and what order I process samples in labs. On some days, I have over 100 samples I need to process, so I want to make sure I take the first collected sample, process it first, because the longer they sit, the more potentially funky the data can get. So, right, the exact time you scooped on that label for me, it's super helpful. On a quick note, we do occasionally ask you to take 2 samples one day. So if you ever open your cooler and you see that there are 2 bacteria bottles in there, that just means I'm asking you to take a duplicate sample. That's just for me to check and make sure that there are no contaminations happening in our lab processes. It's part of the QA/QC, the quality control we have to do. So our data is used by the state to help inform good decisions. So if you are duplicate, I will generally be giving you a warning. When I shoot you your reminder email, I'll say, reminder, you're duplicate. You have 2 bacteria bottles this week. But if you ever see 2 bottles, just take them both. Too much samples is never a bad thing. After you take your bacteria samples, your next step is turbidity. Again, that's like how much sediment is in that water, how cloudy it is. This bottle is not sterile. Imagine it the same size, that 100 milliliter bottle. So it's gonna be about yay big. It's gonna look like a Nalgene bottle. So that kind of cloudy, opaque plastic. These are reused. So sometimes there'll be some old Sharpie marker written on it because someone wrote on it in the past life. I sterilize them between myself. So you can reuse these bottles. They don't have to be truly sterile because all I want to do is make sure they're clean. With these bottles, because we do use them multiple times, we do rinse them 3 times before collecting that 4th sample. So you're still wearing gloves, not like you're worried about contaminating the sample, but because we know the water could be dirty, and so we're protecting you from the water. Wearing your gloves, you're going to kind of find a undisturbed spot in the water. You want to avoid a spot when you were standing or maybe you actually fell in the water before, because we want to make sure that there's no sediments from the bottom of the creek creeping up into your sample. So take a step or 2 upstream, give it a minute for the stream to clear off of whatever you did while doing the temperature, the pH of the bacteria sample. And then similar to the way you filled your bacteria bottle, you're just going to scoop it, getting it all the way full as you possibly can. But this time you're going to dump that water and repeat it 3 times. Scoop, dump, scoop, dump, scoop, dump. Once you've rinsed the bottle 3 times, you'll take a 4th scoop, cap it, and that's the one you're going to keep. You'll fill out an identical label. I'll know which one's your bacteria and turbidity sample because the bottles look a little bit different. And record that time and put it in your cooler for safekeeping. The last thing you're going to do is you're going to take some nutrient samples. These are the ones processed by the Smithsonian. Remember that one of your steps to be fully certified is that you will meet me or a partner at a stream demo at some point. And so these instructions right now feel at all overwhelming. You'll have a manual next to you with a cheat sheet of exactly what to do while you take the samples. And I'm going to break them down really simple when we're in person on a stream. But with these nutrient samples, you're going to take 3 bottles. These 2 kind of blue cap clear, they look like big, like, I don't know. They're called Falcon tubes. Those ones, one is for nitrogen, one is for phosphorus. They're treated exactly the same. So one of them will be, it's just random. Those will be just like the turbidity. You're going to uncap them. You're going to rinse them 3 times. You're going to keep the 4th, put them in the cooler for safekeeping. Notice that they do have the little labels on them. We've updated labels so they're a little bit crisper looking this year, really similar to the bacteria and the turbidity labels, making sure you have the date, the time, location written on them. Your 4th bottle is one that we're going to use for an organic, looking for organic concentrates in the water. And this one has to be filtered. So that means we're actually trying to remove the bigger particles and really looking at dissolved organics to do this one. And so it does need to be pushed through a filter. So in your kit, you'll have a syringe. It's a 60 ml syringe, much bigger than one you'd ever use for medication. And also a filter, this small little oval thing that screws on the cap of it. So the thing you need to remember is everything before you want it finished needs to be rinsed 3 times. And so you'll rinse the syringe 3 times. You'll then draw some water and just push 3 little bursts of water through the filter. So the filter has now been rinsed 3 times. And then you'll take some filtered water and rinse the brown bottle 3 times. That should take about 60 mLs of water to make sure you rinse 3 squirts out of your syringe, rinse 3 squirts through your filter, rinse 3 squirts through your brown organic carbon bottle. Next, you'll just take the filter off because you can't draw water through the filter, fill the water, screw the filter back on. And I will say it takes a good bit of wrist power to push the water through the filter. The dirtier the water is on that day, especially after rain, there's a lot of suspended sediments. Sometimes you're going to get yourself a workout pushing that through. Ideally, you'll be having 100 milliliters. That would be 1.5 syringes full of water that you're going to push through the filter into your brown bottle. But my rule of thumb is do what you can do until your hands give out on you. Make sure everything is labeled. with your station name, right? That'd be the LB1, the date, whatever day you're out sampling. So look like May 15th, I'm going out, and write your initials. All that duplicative information, just make sure I know exactly where that bottle comes from. And again, we'll go over these procedures in person. I feel like they'll be a lot less scary when you're in person with me. The last thing though is that every once in a while we're not doing this at every site, and we're only doing this once a month. So it's possible you'll never have to do this, but it's possible you will. So I'm giving you the information. We'll be grabbing some bigger Nalgene bottles for something called microbial source tracking. As this project centers on E. coli, we've built this 5-year database of how much E. coli is in our streams. But the missing gap is, where does that E. coli come from? And so microbial source tracking is a really technical thing that I don't quite understand what happens in the lab, but they're able to take those little E. coli's and speciate them, because different species of that E. coli is correlated with different types of animals that the fecal bacteria came from. So is this goose poop E. coli? Is this dog poop E. coli? Is this human E. coli? And so this is a really exciting project for us, because it will allow us to actually understand where the E. coli is coming from, and therefore actually do something to start to reduce E. coli levels. So far, we've just tracked it. So moving towards action is really exciting for us. If you are told that you're an MST site in a given week, you'll find 2 extra Nalgene bottles for you to travel with. Those Nalgene bottles are sterilized, so please do not open them till you get to your site, and your glove with new fresh gloves. I'll make sure you have enough pairs to switch gloves before this step. And then again, this is so easy process. You uncap the bottle, you scoop water into the bottle, you recap the bottle. That's all it is. You'll have 2 of them, so you'll do that twice. And then that will allow our partners at UMD, Amy Sapkota and her coworkers, to tell us where bacteria comes from. So we're so excited about that. You will have a field sheet to help you record data as well, to keep track of your procedures and processes. So you'll write your name. This is actually one of our DC sites, but your Maryland sites look identical. You'll just write down, again, Long Ranch and Rolling Terrace, LB1, the date. You'll write your name, first and last is great. Just first, I can figure out who you are, what time you monitor at. It could be a range of time, like I was here from 9:15 to 9:32. That's perfectly fine as long as I have one start time there. You have a few observations you'll make. These are just kind of more qualitative ones. You'll circle the one word that feels right, feeling free to add as many notes. Write me a story, write me an essay, helping explain why you circled that one thing. So was the water surface calm? Were the ripples? Maybe you're at Bladensburg, there's white caps rolling across the river then. Circle 1. Stream flow can be a bit funky, especially if it's your first time at the site. You don't know if that's normal or not. When in doubt, circle normal, but if you feel like it's low, if you feel like the water's not moving very much or it's still, you can circle low or stagnant. If you feel like it's racing on by, go ahead and stick high. Weather type, watercolor, you're using on normal, abnormal, remembering that water in streams should not look like water coming out of the faucet. So various browns and green hues are natural. If you feel like you want to circle abnormal, it means that you think this water something is horribly wrong with it. So while you circle abnormal, please also hop on the phone. You'll have my phone number and call me and say, you have Maluchin. Please come ASAP, because this is horrible. If you don't feel like you need to call me because you don't think it's that big a deal, it's probably in the normal family. And then you'll just circle if you see any of these observations. You may circle multiple at this point. You see any oil slick. SAV is submerged aquatic vegetation. It's a good thing. Dead fish erosion, foam debris. Again, if you feel like anything's alarming, please call me right away, because I would love to help flag people and get the process moving of abating that pollution if it's occurring. Record your data, a little checklist to make sure you did all your steps. And then on the back side of that, you will have a chain of custody form. This is a legal form. If we ever needed to use this data as part of any legal movement, if there was any pollution we wanted to report, take legal action against, this just proves that data was collected, and those samples were cared for since collection through processing. So your name and signature at the front saying, yes, I took this sample. Here's my signature. Every single line will refer to a bottle. So this person looks like he went to a couple sites, also had a duplicate, so took 2 bacteria at one site. For every bottle in your cooler, you'll have a line. You'll write down the sample ID, the type, if it was bacteria, turbidity, nutrient, whatever. And then when you turn it into the person, whether it be me in the lab in DC or one of our partners up in the drop-off spots, they'll sign it just saying that they took custody or ownership of that bottle. Before you leave the site, make sure everything is labeled. One last check of your data sheet and labels. We don't want to have any blank spots ever. Make sure everything is in the cooler. Make sure the cooler has ice in it. Make sure that the bottles are sealed and upright. Check your field note sheet, your chain of custody. Again, no blanks. We filled everything out as possible. And then please do not leave trash. We know that sometimes we have little pH strips and maybe some of the wrappers left over from nutrient sampling. Just shove your trash in the cooler. I will throw the trash in the lab. Please do not leave it at the sites. Take a picture. Once you start following us on social media, you'll see that we do share the data alongside pictures of volunteers. So I love a good smiling picture of someone doing a sample, whether it be a selfie or it be a picture if you have a partner or friend out with you, that'd be lovely. Even just a picture of the site so I know what it looks like, because I like to see how they change in the season. I don't get to go see the sites all the time because I'm stuck in the lab. Take a picture. And then you'll have to drop off your sample. You have a few options of where you'll bring that cooler when you're done. One of our amazing partners, Superstar Monitor Deb, will be at Tacoma Piney Branch Park. That's not the Piney Branch Tacoma Middle School. It's adjacent to it near a dog park. There's a pavilion there that she'll be sitting from 8:30 to 10 at 2 Darwin Ave. So you can drop it off there with her. You can also drop it off at Joe's Movement Emporium. They have been graciously volunteered their space. They'll have staff there between 8 and 10 in the morning. So you have to be there. They will not open until 8. Around 10, I'll be there to grab the sample and bring it into my lab. So have it there by 10 at the latest. And that's in Mount Rainier on Bunker Road. If for any reason you're not going to make it there by 10, you want to do a little bit more of a leisurely sampling later in the day, you are allowed to sample all the way up until 11 or even noon. But remember, at that point, it's your responsibility to deliver that sample all the way down to my lab, which is on 8th Street. That's the Navy Yard across the barracks in Washington, D.C. You won't do this, but it's fun to know. Once I get your sample, I will process it. So I will mix your water samples, that 100 milliliters that was in the bacteria bottle, with something called a collar. It's basically like food for bacteria. But the fun thing about this food is when a bacteria eats it, they glow. They become fluorescent green. And so the more they eat, the more green they become. I'll put that in an incubator, basically an oven that sets a body temperature, and it will cook in there for 24 hours. Then I'll pop it under a UV light. And again, if there was E. coli in those cells, they ate that nutrient, green glowing collier, which is really expensive, and I don't quite understand how it works. They will now be glowing bright green. When I count the cells and do some fancy math, I'll be able to estimate how many colonies of bacteria were in that 100 milliliter bottle. If there was under 410 colonies of bacteria, EPA says it's safe to recreate. So I'll go ahead and let the public know that site passed for that week. If that count was over 410, looking at this tray I have, it would be well over 410. That one's going to be over 1,000 most likely. I'll go ahead and put a big red on that site, shouting out the public, hey, you might want to go splashing. Keep your kids at a Long Branch for the week. Turbidity, I also processed one in sample. This one's pretty easy. I have a nephelometric turbidity unit that does all the work for me. I just transfer your big bottle. I do this multiple times because with science, you always replicate things. But I'll transfer it into a small glass vial. And in that small glass vial, I'll put it in the machine. The machine shines light through the sample and has a sensor to see how much light comes through it. If all the light comes through, it's perfectly clear. It's a beautiful 0 turbidity. But the less light that travels through, that means it's bouncing back off some suspended particles. It will increase the turbidity that was measured. And so if that machine reads anything under 20, turbidity is good. I would much prefer it from a stream health perspective to be a little bit closer to 15, but up to 20 isn't going to be unhealthy. If it's above 20, it rarely is, except for unless we had really extreme rain events. But that's another reason why I might flag a site in my weekly social saying, hey, this site failed. Please don't recreate it in the coming days. Nutrients, I don't have how that's processed because that's extremely complex. Nutrient data won't be coming out week by week. That's a much more complex, more batch cycle. So Dr. Hale, who works at Smithsonian, will be presenting her results in the end of the year for you all. And the UMD, the processes they do in terms of MST, I know that they're isolating the bacteria. They are then genetically coding the bacteria, comparing it to known libraries of E. coli found in various species. They'll also be presenting their results towards the end of the summer once we have results to present. But they will not be seeing those weekly because those processes are a lot more complex than the ones I do in my lab. It takes me 24 hours for those samples to incubate to ready to share out data. And so the big thing I do is push it out on social media. So please make sure you're following Anacostia River Keeper or some of our partner organizations, as we will make sure this data gets posted weekly so you know what results happened. I'll also email them to you if you're one of the people who monitored that week. I also post this data to Swim Guide. If you don't have Swim Guide, it's a great app to have on your phone, especially if you get out and travel in the summer. A lot of river groups like mine will post their data. So if you're out near Annapolis and you want to know if Sandy Point is a good place to swim on a weekend, the app will tell you. We also upload our data to the Chesapeake Monitoring Collaborative. It's a large group of scientists. That's a little bit more of a scientific place, but if you're looking for long-term data, you want to download multiple years worth of data, that's where I'll point you towards. We also host it on a map and on a database on our anacostiariverkeeper.org website. So that's what I have to do for the update and the training today. I know that I sped through procedures a little bit, and that's because I do have one more step for you to do, actually 3 more steps for you to do before you're fully certified. So in the same email that you probably found the link to this video, or on the QR code, you will find the certification test. It's an open note quiz, so please feel free to go back and refer to the video, watch it again as you take that quiz. We're just looking to see that, make sure you pass some basic things, you know how to properly take the samples. It's just a QA/QC thing we have to do to have our data. Once and while you're in that certification, you're going to sign up for a stream-side demo. Depending on when you watch that video, there might be some options. I try to keep those updated on when I'll be at different places. You will have to join me. It'll be about a 10 to 15-minute time when me and you hang out in the stream. We'll pretend to collect the sample together so I can show you some of the intricate details that were hard to see on the computer screen and sign off that you are officially certified and you know how to do the sampling process. There also is a waiver. Like everything these days, the world has to have waivers. It's just a basic standard liability waiver. It is on the QR code. My head is kind of cutting off, but will be also found on the email you found this link on. Once all those things are completed, you'll find we'll just give you an email letting you know that we have done counted all your steps. You're a fully certified volunteer. You'll be added to our volunteer email bank, giving us weekly email shout out with the results, as well as weekly links to the signups. Again, you can sign up for once and just do one sampling effort, or you could sign up every single week for the rest of the summer. Thank you so much for listening to this call, and I look forward to meeting you and seeing you out in the Anacostia watershed.